All around us, there's this entire world of information that we just can't see. I'm talking about invisible chemical changes happening all the time that could tell us if our food is still fresh or even if a disease is starting to take hold. So today we're going to dive into how we're teaching machines to see this hidden, invisible world.
I mean, just imagine this for a second. What if you could know for sure that your food was starting to spoil days before you could see or smell anything wrong? Or what if we could pick up on the tiny chemical signals of a disease long before any symptoms actually show up? This isn't science fiction, you know. It's about learning to read the chemical clues that are literally all around us.
We rely so much on our senses, but let's be honest, they have their limits. The really critical changes, the ones that signal decay or disease, they all start on a molecular level, a place that's completely invisible to the human eye.
Think about it, right? We judge a tomato by how red it is, but that's a super incomplete picture. That slice of bread on your counter might look perfectly fine, but on a microbiological level, decay could have already started its work. Our eyes just aren't built to see that. So, how do we get around our own biological limits?
Well, we do it by creating a whole new kind of sensory system, but for machines. And it works in two main parts. The best way to think about it is like this. We're giving a computer a set of incredibly powerful new eyes to gather information, and then a really sophisticated brain to understand what it's actually seeing.
So the eyes. This is a technology called spectroscopy. To put it simply, it shines a light, could be visible light, infrared, even X-rays, onto a sample. Now, every single chemical compound out there interacts with that light in its own special way. It absorbs some of it, reflects the rest, and this interaction creates a unique signature, kind of like a chemical fingerprint.
It's a really elegant process when you break it down. Step 1, you shine the light. Step 2, the molecules in the sample do their thing. They react. Step 3, a detector reads that resulting pattern. And that result is the fingerprint, a raw stream of data that tells the story of what that sample is made of chemically. But there's a catch.
Okay, look at this. On the left, that's what we see, a totally normal slice of bread. But on the right, that's what the spectrometer sees. It's this messy, super complex graph of raw data. And to us, this graph is, well, it's meaningless. It's just pure information without any interpretation. The eyes can see, for sure, but they can't understand. And for that, we need the brain. And this is where chemometrics comes in. It's the brain of the whole operation. Chemometrics takes that huge, messy data set from the spectrometer and throws some powerful machine learning algorithms at it. Its job is to sift through all that noise, find the hidden patterns, and translate that chemical fingerprint into something we can actually understand and use.
So this all sounds great in theory, but how does it actually work in a real-world scenario? To make this super concrete, let's look at a brilliant experiment researchers ran to predict food spoilage. We'll just call it the bread test.
The goal here was really clear and pretty ambitious. Could they combine the spectroscopic eyes and the chemometric brain to build a system that could warn us about bread going bad long before our own senses had any clue?
And the stakes for this kind of work could not be higher. Foodborne diseases cause an estimated 420,000 deaths every single year. A huge chunk of that comes from food spoilage. So detecting it early isn't just about saving your sandwich. It is an absolutely critical issue for public health and for global food security.
So here's the play-by-play. They took pictures and spectral data of bread over a 10-day period. Now, look really closely at day 8. To the human eye, that bread still looks totally fine, but lab tests, the ones that count the actual bacteria, show it has already crossed the line into early spoilage. This is that invisible window of opportunity the machine needed to find. So the eyes collected a ton of data. We're talking 48 different features, from color and spectral info for every single reading. But then the brain, the chemometrics model, it went to work. It didn't just look at all the data blindly, it did something called feature selection to find the 15 key signals that were the absolute best predictors of spoilage. It automatically figured out what truly mattered. And the results? They were incredible. They tested a few different machine learning models. The best one, a model called XGBoost, was over 92% accurate at correctly identifying if the bread was fresh, in early spoilage, or totally spoiled, based only on that invisible chemical data. That is just wow. This quote from the research paper just sums it up perfectly. This is not just some academic exercise. It's about creating practical, real tools that help us cut down on waste and stay healthy, all by acting on information that, until now, was completely invisible to us. And as amazing as the bread test is, This powerful combo of spectroscopic eyes and a chemometric brain has applications that go way, way beyond the kitchen. I mean, this same fundamental idea is starting to revolutionize tons of fields. In medicine, it's being used to find chemical biomarkers for early disease detection. It's helping design better drugs. It's analyzing forensic evidence. It's ensuring the quality of pharmaceuticals. It's even identifying specific types of bacteria. It is a true game changer across the board.
So, what's the really crucial takeaway here? It's this. By combining spectroscopy and chemometrics, we haven't just built some new tool. We've effectively given our machines a powerful new sense. It's like a kind of chemical vision, and it's allowing them to perceive the world in a way we never, ever could.
And that just leaves us with a really fascinating question to chew on. Now that we know we can teach machines to see the very chemical building blocks of our world, what other invisible problems are out there just waiting for us to finally see them?